Nope, not want that.
Hi, Anu, Cameron. Welcome to the physics poster session. Aman will go ahead and get started soon.
Hello, everyone. I'm Aman Patel. I'm a junior bio major, and I work in a quantum optics lab with Dr. Zhang in the physics department. It's a lot. It's a lot of big words, but mostly it's just me shining lasers on stuff, and it's a lot of fun. So, we work with a lot of nanomaterials, and we use Raman spectroscopy to analyze these nanomaterials. And this particular nanomaterial, they're envisioning to use in a breathalyzer for diagnosing diabetes. So diabetic patients, they produce a high amount of acetone in their breath when compared to non-diabetic patients. So 1.8 ppm versus the 0.8 that non-diabetic patients like us would produce. So that difference is large enough to use such methods to detect diabetes in patients. And this would be used to produce a breathalyzer test. So like they do for alcohol, right? There's those breathalyzers. They analyze the alcohol content in your blood based on whatever feedback that they're getting from your breath. Same way, we are envisioning to use this nanomaterial to make a acetone breathalyzer that monitors diabetes. This would provide a much more accurate and non-invasive procedure to monitor diabetic patients. So a lot of nanomaterials have been tested for their efficiency of detecting acetone in gaseous form. But this nanomaterial, or potassium tungsten oxide, has had the most success in terms of how low of an acetone content that it can detect and interact with. So how this works is the material is a semiconductor. So when acetone passes over our material, it deposits electrons on the material and has an electron transfer. This electron transfer causes a change in its resistance. So it increases in resistance based on how much acetone is passing over it. And we can use that difference to measure out how much acetone is present in that gaseous form. So we use Raman spectroscopy to analyze this interaction between the two materials. So if we understand the actual mechanism of what's happening, we can enhance it and we can use it to make it more sensitive and make it ready for a marketable product. So this is a very simplistic diagram of a Raman optical microscope. So what we have is we have a laser, and the laser is, don't worry about the mirror and all that, but mostly what we do is we take this laser and we use it to excite our material and our electrons of the material, and whatever feedback that we get from that material is what we put into a spectrometer to give us the spectra that you see as my results on the right. So what Raman spectroscopy does is it's Raman scattering versus photoluminescence. So the difference is a little difficult to explain. So in solid state physics, which is a topic here, it's solid state physics. So there's something called the Fermi level. And at the Fermi level, just imagine like it's the highest number of electron energies possible. So think like the very outside of your orbits. And you have your empty electronic orbits and your full electronic orbits, right? So E naught. So this is our full electron orbits, and it's called the valence band. And this is called the conduction band. Now, the structure here for each material is unique, and it determines its electronic properties. So in Raman scattering, what we do is we have something called virtual state. So if you think about those electron levels from like chemistry, right, N1, N2, N3, so there's little spaces in between them. So what Raman scattering does is it excites the electrons to these virtual energy states, and then as the electrons get back to their initial state, they give off radiation that we see as spectra here. And they give off radiation because of their changes in energy. So there's 3 different types of Raman spectroscopy, but that's a little more difficult to explain. Versus photoluminescence. So luminescence, you're exciting your electrons to a real energy state, which is our conduction band. And when our electrons get back down, they release a photon that we observe as photoluminescence. So we aim to use our Raman scattering to analyze what is happening on the surface of our material. So I use what is called a Linkum cell. In a Linkum cell, it's essentially an isolated environment, so it controls the amount of gas and what is present in there. So I put my sample in our Linkum cell, and I'm passing gas over the material. So first to get a baseline, like as an atmosphere, and then nitrogen. So that is to acquire a baseline Raman spectra. because they're present for all materials and they're unique to them. But when we have it interacting with something, there's shifts in the Raman spectra, and that is how we tell if there's some sort of reaction. So what I do is we have nitrogen pass over, and then once I have my baseline, I add in acetone with our nitrogen, and the combination passes through and interacts with the material. Oh, sorry. So if you look at the top two graphs here, it's not letting me zoom in for some reason. Oh, there we go. Nope, not. Oh. All right, so if you look at the top two Raman spectra that are present, so I have two samples. One is the sample kit, the potassium tungsten oxide with a surfactant, and one without. So this was to see that there's no difference in Raman spectra caused by our use of our surfactant, because it is only aimed to increase the intensity of our material so we can better see what the interactions are. But continuing from that, so on the left is our material with our surfactant, the right without. As you can see, we observed no real shifts in our Raman spectrum as we were going through our experiment, because nitrogen plus acetone, the only thing we see is the decrease in intensity, and that is due to the presence of acetone over the material and its interaction with the laser path. But what we did observe was this little rise at the very end, which you can sort of see. So the Raman spectra and the luminescent spectra exist in different positions. So wave numbers versus wavelength. I'm not going to go into the math there, but higher wave numbers are where photoluminescence exists. So since we saw this rise, we shifted our perspective towards our photoluminescence spectrum. Now, the interesting thing about this was that the laser that we are using is not nearly powerful enough to give our electrons enough energy to have it go to a real energy state and release a photon. So this whole photoluminescence could not have been observed with this laser, but we did. And this was only in the presence of acetone. So this isn't something the material does on its own. It's in the interaction with the material and our acetone. And this tells us a lot about the mechanism of what's happening here. So we observe a very strong photoluminescence peak here compared to without our surfactant, which is expected because of the enhancement. But there is a small peak if you can observe that. Right. So what this does tell us, though, it's a lot about our band structure. It's not letting me zoom in. Okay. Nope. Don't cooperate. Okay. I'll just have to do it from the outside. So like I was talking earlier, right? We have our Raman states and we have our luminescent states. So what happens is there's something called defect states. So the interaction between the materials. I'm sorry, everyone. I don't know what's happening here. All right. So what happens in defect states is when our material interacts with something, there can be the production of defect states, which essentially cause little variations in our band structure. So our conduction band up there, it can have little variations where it comes down to a lower energy state. So what happened is, what we're proposing is that the interaction with acetone causes the change of band structure that lowers our conduction band state. So when we excite our electron with our laser, it is enough energy to get it to our defect state and then cause our photoluminescence based on the data that we observed. So this is everything I have for you today. Are there any questions?
I guess I have a question. So, doing all this, what exactly would you say the next steps would be to the study?
So the next steps would be to go more into test the mechanism more with different concentrations of acetone and test our sensitivity of our photoluminescence, because we have proved that our photoluminescence peak does exist and it is due to our laser in our interaction with acetone. Now it is more into diving into what is the sensitivity to determine the exact energy levels on the band structure, and that's a lot of math, a lot of experimentation that's yet to be done. But the more we understand this mechanism, the more we can optimize it for something for our material. Anyone excited for energy, physics research now, or still completely understand either way? Anyone else have any questions?
So,
Mhm.
would this be for diagnosing diabetes, or would it be for like testing instead of like how you would prick your finger to see your sugar levels? It would be for like testing your sugar levels for a diabetic person already.
It would be for both.
Okay.
So mainly our concentration would be to make it easier to diagnose diabetes, but this can also be, again, the sensitivity can be increased, and if it is, we can use it to monitor diabetes in diabetic patients. That way they don't have to prick their finger every time for their glucose levels. But yes, it can be applied to both.
What is that last graph with dependence of environment saying? Like the ratio depends on what chemicals you put in it?
Yes, so those are the different various gases that I passed over the material. So first, our main focus was on the strongest one that you see, which is our acetone and our nitrogen gas. Nitrogen gas doesn't have any involvement with our material. It's inert in the scenario, and they don't have any interaction, so it acts as a good baseline. And we have to use a carrier gas to get it through our Lincoln cell because we can't just pump acetone as a gas in there. So what we do is I use oxygen, nitrogen, and then ethanol as to test it with a different solvent to see the difference in sensitivity. So it does interact with ethanol as well, but the difference in, as you can see in the ratio, the difference is so high that we don't have to worry about that in a real world application. We also tried it with oxygen where we have a very major decrease in our intensity, and that we propose is due to the capabilities of oxygen and its ability to pull electrons from other things. But yes, that is just to test our material against other gases as well.
I'm guessing there isn't, but is there any variance in the acetone concentration in diabetic patients' breaths and according to what type of diabetes they have at all? Or
I have not read any literature based on that, but I cannot say much about that. I don't know if it's varies due to diabetes, but mainly the results that we're going off of is related to type 2 diabetes research, where they found this.
well, thank you. I'm linking the sign-in sheet in the chat for everyone to fill out. Yeah, thank you so much. I mean, that was a really great presentation.
It was interesting. Thank you.
Okay, it was really interesting. Yeah.
Thank you.